Kia ora koutou, ko Melanie McConnell aho, he kei ranga hou mati puku puku. I'm Melanie McConnell, I'm a cancer researcher. And my interest, as Kelly said, is predominantly in brain cancers. But I thought I'd just tell you a little bit about me. I grew up on the Kapiti Coast. So this is the prototypical picture of Kapiti Island and the Kapiti Coast, taken from not very far from where I would walk my dog. But the picture that I really like of the Kapiti Coast is this one. You can see here is Malaghan Beach, here is Kapiti Island, and here is the Melbourne Sounds. So we're really not very far from home. I have to say this is my first actual visit into Blenheim itself in the 40-something years I have been on this planet, and I'm really embarrassed. Shame on me. It's very close, and this is actually my first time, but we've had such a terrible summer in Wellington, I'm wishing I had come earlier. So I had a really great science teacher in Kapiti, Paraparango College. His name was Mr. Billows, and he really inspired me to get stuck into science. And so I went to Otago, where I did my bachelor's degree and then my PhD. And so I thought in this audience we might have some people who recognise this. This is where my research career started. This is scabby mouth. This is a viral disease caused by a virus called Orth. The sheep pick up the virus off the grass, gets into grazes and abrasions, causes these quite gory black-green scabs that are full of live virus. This is where I started my research career, and after a mishap in the lab, I decided this was not, virology wasn't going to be the career for me. I didn't give myself an infection, but I came very close. So, gave virology a bit of a birth, and I moved to cancer research. And so, I've worked in all sorts of different areas of cancer research. We've looked at kidney cancer and leukemia and lymphoma and melanoma and breast cancer, but about 10 years ago, I came back to New Zealand from overseas, and I took up a position at the Malaghan Institute of Medical Research. And here it was to work on brain cancer. And kind of naively, I went, blood cancer, brain cancer. It's all cancer, it'll be great, it'll be fine. How hard can it be? Turns out the brain's a really complicated organ, and it's quite tricky. Most recently, I'm now a senior lecturer at Victoria University of Wellington, which is still called Victoria University of Wellington, for a little bit longer. And so I teach undergraduate biology, and I have a research program. And we're really focused on understanding how brain cancer cells, or how brain cells in general, and those cancers in particular, how they survive stressful situations. So I'm going to give you one of the slides from my undergraduate biology class. This is what we think of as being a human cell. So this is a very representative picture of a cell. We've got the membrane around the outside of the cell that holds all the bits in. We've got the nucleus in the middle, which has got the DNA, it's got the genetic information, and then there's all these other organelles and things that make up how the cell functions. And so the ones that we're quite interested in are these things here called mitochondria. Now, how many people have heard of mitochondria? Has anyone seen the meme that was really popular a few years ago? The mitochondria, about mitochondria? Some of you might have seen that one. So, keep that in mind. So we're really interested in mitochondria. And so these essentially are the batteries in the cell. Now this is a representative kind of cell. It's not what brain cells really look like. So if you think about what kind of cells we have in the brain, there's lots of quite diverse cell types. And so we've got neurons, and these are the superstars. Everyone thinks about the neurons. If you've got a neurodegenerative disease, then your neurons are dying. And they make up about half of all the cells in the brain, and that's these really long ones here, and they'll reach down and they'll connect to another neuron. And then these guys, because they're passing electrical signals, they need insulation. And that insulation is a protein called myelin, and that gets provided by the oligodendrocytes, which are these cells here. Does that pointer work? Can you see a pointer? No, okay. These oligodendrocytes are the ones that wrap around the nerves and around the neurons and insulate them. We've got astrocytes. These guys feed the neurons. They take food from the blood and they supply them to the neurons, and they take away the waste. They're the caregivers of the brain. We've got immune cells in the brain. These ones here are called microglia cells. And these microglia are the kind of immune sentinels. They monitor for infection. They're involved if you get inflammation of your brain. And then we've got some cells that make up the blood vessels. And then there's the pericytes that wrap around the blood vessels, and they form this blood-brain barrier. So we know that the brain protects itself by this blood-brain barrier. And so that's the role of those cells. So there's lots of different cell types. And actually they don't look like the picture either. So this is what these cells look like when you find a textbook, but actually what they really look like is this. So this is a human brain cell. It's growing in a dish. So it's growing on a plastic surface. We put some raro basically on top of it. People still drink raro? People know what raro is? Yeah, okay. Some of my 18-year-olds don't know what raro is. So they grow in a dish, and this is a bunch of cells. And so if you look here, this bit is the nucleus. So that's kind of like the picture. It's a round blob in the centre. It's got the DNA in it. And then the edges of the cell are shown out along here. There's another cell here, there's another cell here, and you can see they're reaching out and kind of touching each other. And what we're interested in is the mitochondria. And so all these lumpy bits in here are mitochondria. And we can label those mitochondria with this red dye. So these are the mitochondria, and you can see they're kind of forming these sort of sausage-like shapes. And they're really diverse, they're really active. And so we just watched down the microscope on these live brain cells, and this cell's sitting here, and this is taken over in the course of about 2 or 3 hours. And so the mitochondria just moving around inside the cell. And the mitochondria, like I said, they're the batteries of the cell. And another slide from my undergraduate biology classes. Where did these mitochondria come from? So our understanding now is that these mitochondria were once bacteria that lived outside of the cell. And a lady called Lynn Margulies proposed in the early 1970s that these mitochondria were bacteria. And those bacteria got taken up into a cell, and they made a home in the cell, and they started dividing and living in that cell. And for some reason, the host cell decided it would keep these mitochondria. So, why would you do that? This is the question I asked my secondary cell biology students. Why would you let a bacteria live inside you if you were a cell? Why would you do it? Why would this evolve? Because it's beneficial. You guys are much better at answering questions than 19-year-olds. It's beneficial. There's going to be a functional advantage for these things to happen. So what's the advantage of having a bacteria living inside you? Turns out the bacteria are the ones that act as the batteries. So these bacteria evolved into mitochondria. The mitochondria take oxygen and turn it into energy. And so this is the main role of the mitochondria, with these protobacteria that we adopted about 2 billion years ago. So this led to this phrase in the 1957, the mitochondria is the powerhouse of the cell. And so the mitochondrion is a single mitochondria, or 2 mitochondria is the plural. They're the powerhouse of the cell. Without them, the cell has a really hard time making enough energy. And so we need to have these mitochondria. We need them to make all the energy that cells need. So these mitochondria still look a lot like bacteria. They've got lots of characteristics of bacteria. Their DNA looks like bacterial DNA. So mitochondria, even though they live inside a cell that's got a nucleus, they've got their own DNA. They have their own genomes, and they look it looks like a bacterial DNA. There's bacterial kind of genes in there. They have membranes. The bacteria have a very simple membrane, The mitochondria have this really convoluted membrane. The membranes look like bacteria. And then the bacteria and mitochondria reproduce in the same way. They reproduce by dividing. So there's still a lot of bacterial activity associated with these mitochondria. Now, I borrowed this picture from the evolution department at Berkeley University, so UC Berkeley. And on the same site, they make a statement. And what they say is every new mitochondrion must be produced from a parent mitochondrion in this dividing way. If a cell's mitochondria are removed, it cannot build new ones from scratch. And the mitochondria aren't free-living bacteria anymore. The first eukaryotic cells evolved more than 1 billion years ago. And since then, these organelles have become completely dependent on their host. So this is fact. This is the dogma. This is what they say at Berkeley. It must be true. This is what everybody believes. There's no reason to think that that's not the case. Mitochondria do a few other things. They can initiate death. If a cell gets injured, the mitochondria can be the suicide gene. And so this is what happens if you injure a cell and you injure its mitochondria in particular. If you injure the mitochondria really badly, you're going to take a cell from looking like this, nucleus, and the cell will spread out to these dying, we call them apoptotic cells. Mitochondria are also very topical. They have a role in sex and gender and aging. I'm not sure what the role is in gender, to be perfectly honest. Gender is a social construct, so how the mitochondria are involved in that. There's a lot of sort of evidence that mitochondria have a really wide-ranging role. If you're interested, there's a great book, Power, Sex and Suicide, about mitochondria. So, we all know who Mr. T is. Yes, he's a powerhouse. So this is where we get to what I call the heretical mitochondrial biology. If Berkeley says it in their literature, it must be true. Mitochondria are totally dependent on the host cell. They can't reproduce themselves. They can't, a cell can't renew from scratch. But we know that that's not true. The data from our group and from others around the world is starting to show that what we've believed to be true all these years about mitochondria is just wrong. So this first report came out in 2006, so it's 14 years ago now. And what they said is that mitochondria can move from one cell to another cell, and it's doing that when the recipient cell has got a defect in its ability to make oxygen, to respire, to produce energy. And so this was the first hint that mitochondria maybe weren't quite as docile and domesticated as we thought. And this paper completely just disappeared. This particular group worked in Tulane University in New Orleans, and Hurricane Katrina hit their lab about a month after they published this paper, and their lab was destroyed. They lost everything. Everyone had to move, and so this research finding just disappeared. No one read the paper, no one saw it, no one followed it up. And so about 8 years later, when we discovered the same thing, and we thought, 10 years later, we thought we were the first people to find it. It turns out we weren't. But one of the things that we know now, and it's taken a while to convince the scientific orthodoxy, is that mitochondria can in fact move between cells. So, they are not totally dependent. And what we've got here, and this is the first of the red-green pictures, this is human astrocyte cells, and what we've done is we've labelled half of them green and half of them red, and we've just put them in a dish together, and we've just put the microscope on and we've watched, and we've just taken a movie of how these brain cells interact with each other. And so we have some mitochondria are green and some are red, and you can see when you look at this that there's a red spot inside this green cell, this green spot here inside these red cells. And what I want you to do is play this movie, and we're going to watch this guy here. This is a cell that has green mitochondria, and we just watch these for 5 hours. So this doesn't happen fast. So we're going to watch this guy. Oi! Go back. We're going to watch this guy here. And he's going to extend this arm down. He's going to extend his arm down. He's going to extend down. He's going to bypass these 2 red cells here. Make contact right down here with this red cell. And then this piece here has been snapped off. So this guy sent a long arm out with his mitochondria in it, snapped it off, and deposited it on the top of these other cells. Now, if the student had not gone home and he'd kept this movie going, we would have been able to watch these mitochondria move into this cell. He told me it was 2 a.m. and he wanted to go home. Okay, fine, it was 2 a.m. and he wanted to go home. If we watch that movie again, there's another example of this happening. So this guy here, going to see it again, send out these really long arms. Now we don't know why they're doing this. We don't know why this guy is reaching out this really long arm to make contact with this cell when it bypassed all these other ones along the way. So we don't understand what's happening here, but there are mitochondria in this long arm, and they're going to be given to that cell. We see this happening. We know that it happens. I've played this movie all around the world to try and convince people that this really happens, because people have said, we don't believe it, it's not true. It can't be true. We've never heard of this before. We don't believe it. And so I've played these movies all around the world, and people just sit there and go, wow, but why do they do it? And I'm like, I don't know. Well, how do they do it? How do they decide which cell? I'm like, I don't know. So we have lots of questions. This is one of my favourite pictures. We captured this. These cells are reaching out, touching each other, and you can see we've got green coming down from this cell in here. We've got red tracking back up this way. So this is a two-way interaction, which makes me think of one of my favourite pieces of art, the creation of Adam. It's the finger of God coming down, and just it's that life force. Michelangelo knew what he was on about, I think, when he painted this picture, because this is what we see going on in ourselves. So it's pretty heretical stuff. People didn't believe it. And so in our cancer work, we showed that if you've got a cancer cell and you have a defective mitochondria, that cancer cell cannot grow until it gets some healthy mitochondria. And the way that cancer cell gets healthy mitochondria is it takes them from other cells around them. And so essentially what that is if your cancer cell has a flat battery, it can't go anywhere or do anything until it gets new batteries. So this strikes us as a really good kind of target for a cancer cell. If you could stop a cancer cell by growing, by stopping it from getting these mitochondria, then that would be great. We need to find out if it's a really important process and if normal cells have to do this thing too, but this is sort of the idea that we're working on, is that these mitochondria moving around is really important. I'm going to skip this one because it's very long and boring. This is the scientific evidence that shows that if you want to be a cancer cell, you've got to have a mitochondria. If you want to be a metastatic cancer cell and spread all around the body, you've got to have lots of mitochondria. At this stage, I was still a cancer cell biologist, but the trouble with New Zealand, or maybe the really good thing about New Zealand, is that we're a small country. And as a cancer cell researcher, I couldn't only talk to cancer people. We're a little country. I ended up in a room with a whole bunch of people at the Neurological Foundation, and they worked in Alzheimer's disease and Parkinson's and Huntington's, and I'm showing them my movies because I'm excited and I want everyone to watch my movies, and they said, you know, neurodegenerative diseases, they've all got mitochondrial defects. Can we do, you know, does this process happen in these sort of neurological diseases? And I went, oh, I don't know. And so we decided we'd find out. So this is the work that the Neurological Foundation funded, that you guys funded with your donations. And so the neurons are really dependent on mitochondria. They really need a lot of energy to be produced. And all of these degenerative diseases, they've all got defects in mitochondrial function. And so we wondered, we knew that brain cells could transfer this because we were interested in the brain cancer. We wanted to know whether normal brain cells could do it too. And then we had lots of questions. Why and how and when and what and where, and those are the sorts of questions that we try to set out to answer. But specifically, what we asked was, if we can stress a cell out and damage its mitochondria, are we going to get more transfer? Is this like something where we can get more transfer in response? Does transfer always rescue a damaged cell? You can imagine if you've got a cell and it's got damaged mitochondria and those go and move to another cell. Remember I told you that a damaged mitochondria can kill a cell. What if you've got cells that have got damaged mitochondria and they're sharing those damaged mitochondria and they're passing on this toxic payload to another cell? So we really wanted to explore this phenomenon in the kind of neuronal cell area. So this is a diagram that my student made when he wasn't on the microscope at 2 o'clock in the morning. And so this is the idea. So we labelled our cells, and again we labelled them red and green because that's the colours that our mice come in. Actually, we have transgenic mice that are red and green, and so we take cells from these mice and we can pop them into dishes. So yeah, we're looking at this idea. We've got a donor cell and a recipient cell, and we're asking how and why and when do these mitochondria move around. So we started out looking at neurons, and neurons are really hard to work with. They're very delicate, they're very vulnerable, they kept dying on us, we couldn't get much progress, so we decided we'd look at the astrocytes. And the reason for doing this is that astrocytes survive and we can work on them, but astrocytes are the caregiver cells. They're the ones that are giving the mitochondria and taking the waste away. So we thought they were a good cell to start looking at. And so we found that rather than just instead of now looking at the initial work I showed you was what we call an immortal cell. It's been a cell that's been in passage for hundreds and hundreds of cell divisions. This is now cells that are fresh out of the brain, and we find that yes, astrocytes can donate mitochondria to other astrocytes. So here is the coloured version of this. We've got mitochondrial donor is in red, and our recipient cell here is in green, and you can see that the cells are reaching out, stretching these arms out, touching each other. We've got green heading up this way, and we've got red coming down this way. So these cells are sharing they're sharing everything. They're sharing whatever we find inside the cell is being swapped. The mitochondria are part of that mix. And so we wanted to focus in really, really closely and see if we could see, is that mitochondria inside the cell or is it sitting on the top? There's a lot of people doing work where they show it sitting on the top, but it's really hard to find it inside the cell. But if we have a look in this picture here, best way to do this. So here's our red donor and our green recipient, and if you look really carefully, there's a little wee red spot here, which is inside this green cell. So we blew that up, really zoomed in on the microscope here, and here is our red mitochondria embedded inside our cell. So we've made this kind of reconstruction of what this looks like. And so we can really see that those mitochondria have got right inside the cell. And so we have no reason to think that they're not functional. We believe that they're going to be actively making energy for this new cell that they're in. So we asked, can we take the batteries out? If we take the batteries out of these astrocytes, will they get new batteries? So we made those cells, we took the astrocytes, we took their batteries out, and the short answer, no. didn't exactly work like that. Was quite complicated. So what it turned out, regardless of how hard we hit those astrocytes, they would not increase the rate of donation to another stressed astrocyte. There's a certain amount of interaction, but we couldn't push them to make them give any more. Didn't matter what we did to the astrocytes, we couldn't make them, give more. There's a certain amount of giving that they're comfortable with, and you're not going to pressure them, they're not going to do anything else. So we found another cell type. And so this turns out to be a mesenchymal stem cell. Now this is, if anyone's heard of stem cell therapy, people have gone and had stem cell therapy, generally this is the kind of stem cell that you're working with if you've got a stem cell therapy. Turns out mesenchymal stem cells are really, really happy to give mitochondria to other cells. And if you stress a cell, they'll do it even more. So, more red and green pictures. This is our astrocytes that were not stressed. And you can see that while there are little red spots around the green cells, they're not really inside the green cell. But when we stressed the astrocytes out, took away their batteries, suddenly we've got this beautiful, I think it's beautiful, this pandemonium, this total chaotic mix of red and green. And what's happening here is it's not like we're seeing one little mitochondria being dropped off into these cells. What we're seeing is hundreds and hundreds of mitochondria going from the stem cells into these brain cells. And so they're not just depositing teeny tiny little astrocytes through a little tube, they're giving all of their mitochondria. The entire mitochondrial network of the stem cell has ended up inside the brain cell. And it turns out that the stem cells are actually fusing. They're physically combining with the astrocytes and giving up all of their mitochondria to these astrocytes. And you can't help but anthropomorphisize a bit here. So the astrocytes are quite takey sort of cells. They'll give if they have to, but they're not really keen on it. Stem cells, really altruistic. They give up their whole identity to fuse with these brain cells. So whatever that astrocyte is doing, it's a really powerful sort of signal that can make a cell just devote itself to keeping this other cell alive. So essentially, what we've been able to show is that these mesenchymal stem cells can completely just give themselves up to these astrocytes. If an astrocyte is stressed, it will take everything that stem cell has to offer. We've got lots of questions. We don't have a lot of answers. We've got lots of questions. So what other kinds of stress might induce? We took the mitochondria out, but we've been looking at what happens if you have an Alzheimer's mutation or you've got a Parkinson's mutation and you can injure the mitochondria that way. What happens if you're having chemotherapy? One of the side effects of chemo is that you injure your mitochondria. Can we get mitochondrial donation to rescue that kind of stress? Mesenchymal stem cells, people pull out of fat or they pull out of cartilage. Are there cells inside the brain that are naturally there that we might be able to kind of rev up to get them to do this kind of rescue process naturally? And there are cell types in the brain that can do this, and so maybe we'll be able to use those in a kind of a therapeutic approach. And then the question that I often get is, can we use stems? Does this mean that stem cell therapy for the neurodegenerative diseases might actually be a viable option? And I think the answer to that is not yet. We don't really understand how this works, but I think that gives us the potential for trying to see how we can rescue these cells under stress. Now, I sit in my office and I write things. These are the guys that did the work. This is Ruby Schneider. He was the one on the microscope till 2 o'clock every night. He was a bit of a night owl. He assured me he didn't want to be in the lab at 9 in the morning. He wanted to be there at 2 a.m, so I didn't upset him too much. Did a lot of this work involved a lot of analysis of images. We had huge big data sets, a lot of computer work, and then Matt Rowe, a PhD student who was funded by the Neurological Foundation as a Miller Scholar. And so he has carried on with this work and he's looked at this in a cancer context. And he's just finishing his PhD now, supported by the Neurological Foundation. So, these guys are part of a bigger team. And so before I finish, just thank the whole team. You never know where your ideas are going to come from, and you really have to be very open to hearing new ideas and kind of believing in the impossible, crazy stuff. So these are the people that give me my crazy ideas.